Thank you.
Got it. Okay, so I'm going to tell you a little bit about the economics of several carnivorous ant societies, and then conclude with a third chapter about some of the myrmecophiles that cause trouble for many different types of ants. So there's a number of apt metaphors that one can apply to an ant colony. Of course, the superorganism metaphor. One of my favorites is the idea that an ant colony functions as a factory within a fortress. That is, the colony itself is a source of production of new workers, queens and males, and it lives inside the structure that it constructs for itself or excavates, the nest, right? And when we think about the way that these colonies function and the fact that they're perennial societies, we have to consider sort of two levels of organization in addition to the extended phenotype, which is the nest, right? We have to consider the traits of workers that live within the colony and turn over quite rapidly. Workers are, of course, short-lived. They're functionally sterile in many cases, and they often specialize on a sequence of tasks as they age or with respect to their morphology, right? So when an ant comes out of its pupa, it begins its life in its career path as a brood care worker, and then it moves up in space and in tasks to perform ultimately a foraging role outside the nest, which is the most dangerous and final role that an ant will perform. Most people who know about ants know this all too well. Of course, colonies are perennial. They are long-lived. Some can live decades. And that includes the sort of germline of the colony, the queen. These colonies actually reproduce, and their reproductive rate and their annual and lifetime fitness is sort of a function of the organization and traits of the individual workers that make up the colony, how labor is divided, how flexible these workers are, right? And so together, these things come together to give us sort of phenotypes at different levels that can be studied. And we can look at the interactions and consequences of organization at all of these levels. To really understand how an ant colony works, we have to understand the way that it produces on an annual cycle, okay? So every ant colony is faced with sort of a production problem on an annual scale. An ant colony must first grow and then reproduce. This is sort of true for every organism, right? So like a typical organism, an ant colony has an ontogeny where over a period of years, it adds workers, the sterile female caste, and then increases in size until it gets to a certain threshold, a certain number of workers at which the colony will begin to reproduce in a specific season. So for the ants shown here, Pogonomyrmex badius, colonies need to contain at least 700 workers before colonies will reproduce in a given year. Now, the reproduction is not the production of workers, right? The production of workers represents growth in a colony size. It is the production of sexual females and males, these alates pictured on the right, which represent the colony's fitness. So this is the actual product that a colony is attempting to produce and by which selection is acting on all the traits of workers and their organization, right? So one of the interesting things we can think about when we think about these ant societies and their annual production problem and their perennial production problem, because they live so many years, is how they overcome sort of the costs of worker mortality. As I said, workers are short-lived and they turn over rapidly. Sometimes there can be multiple generations within a single year. So how can a colony predict the mortality rate of foragers or the workers that leave the nest and die afield? How is a colony able to tune its demography and sort of production rate to replace those workers in order to maintain its size and even to grow? Because we know that colony size is very important for the threshold level at which a colony reproduces. And then beyond that, growth in colony size or a larger colony size can have greater dividends, right? So this is sort of like the idea of economy of scale. In some cases, when a colony gets larger, it can affect the sex ratio, number, and quality of the sexuals it creates. So on an annual scale, a colony has to deal with the loss of multiple generations of workers, the replacement of those workers, and the maintenance or growth in size, which will affect its ultimate reproduction. So how do colonies actually do this? how do they tune a whole population? If we think of the workers in a colony as a population, how do they tune that demography to meet the extrinsic causes of mortality and the extrinsic rate of mortality that these workers suffer? I'm going to talk to you about a couple case studies. First, I'll speak to you about Veromssor pergandei and some of the ways that they overcome production costs and sort of this equation on an annual scale to balance their annual budget in terms of worker birth and death. And then I'll talk to you about my graduate work on Pogonomyrmex badius in Florida, which overcomes the similar problems in a completely different way. Finally, I will talk about myrmecophiles, some of the creatures that exact costs on this production line, and how colonies sort of overcome some of these issues with unwanted guests. So let me introduce Veromssor pergandei. This is a beautiful seed harvesting ant. It also smells really good. It smells like benzaldehyde or bitter almonds. It lives in the Sonoran and Mojave deserts of the southwestern United States. And as you can see, the ants are actually, the workers within the colony are actually polymorphic. They come in a full and continuous range of sizes, with the largest having a head width almost 4 times the width of the smallest. So there's a quite vast variation in worker size within these societies. Okay, so not only do workers sort of divide labor based on age in a colony like this, they may also divide labor based on morphology, as we know very well from things like leafcutter ants, right? So to begin to ask how a colony like this, these are massive colonies. When you go to the desert, you can find them in the early morning foraging in great black ribbons across the desert floor, as thick as your arm, with thousands and thousands and tens and thousands of foragers going out every day. And of course, these foragers represent just a small proportion of the whole colony. So what is the sort of production rate the colony needs to operate at in order to replace foragers that die afield and that will never return to work again, right? How do they maintain their size, especially in a colony of this scale? Well, I want to challenge you to think about colony size in a different way, in a way that hasn't really been spoken about in the ant literature much before. And this is to think about colony size as more than just sort of a snapshot in time. So if we excavate a nest, we can calculate all of the ratios and count everyone in the nest and the sort of whole demography of the colony, larvae, pupae, adults, queens, right? We can do all of that and say this colony is this size. But if we want to know how it maintains that size, we actually have to look at how large it is on an annual scale and how many workers it produces through time. So I did exactly this with Veromssor pergandei and followed a set of colonies across several annual cycles by repeatedly measuring the number of workers they produced in the field. And how do you do this? You might be wondering. You can actually consider the fact that because ants go through sort of this sequential change in jobs, where the youngest ants are contained within the nest and the oldest ants actually leave the nest to forage, one can actually get a pretty good count of the number of workers the colony produces simply by studying the forager population. And that is because every ant born in a colony will become a forager at the end of its life. And so if one were to repeatedly mark and recapture and measure the forager population size in a colony, and do this throughout the year, one could add it all together and determine how many workers were actually produced and made it to foraging within individual nests. And so in order to do this, I worked with a great undergraduate, Yoshida Chavez, who was really up for counting ants with me. And we went out to the desert and picked several sets and neighborhoods of colonies. And we performed multiple mark recapture events on these colonies throughout 2 annual cycles. So what we did was we went out there, we captured as many foragers as we could, operationally defined as individuals a couple meters from the nest carrying seeds and carrying on foraging. Then we quickly placed all of those foragers in a tray, photographed them, and then we were able to spray paint them with fluorescent ink, which has been a staple of most of my research studies. This ink is fantastic because it does not change the lifespan of the ants, and it does not change their chance of mortality in the field. Once you mark the forager population, you can release it, come back the next day, and look at the proportion of individuals recaptured to determine the total number of foragers in a colony that are allocated to foraging. So this is called the linked index mark recapture method. Most ecologists will be familiar with it, but we can apply it to populations even within an ant colony, right? And so when you do this sort of mark recapture work, what you find is that you can get more than 60, sometimes more than 93% of the ants back on the second day. So this is a really like conserved population of individuals in the nest that is very easy to follow over time. So once I did that for a number of nests, I found something quite interesting, which is that regardless of forager number or forager population size within a colony, all foragers sort of had a maximum foraging tenure of about 18 days. So every 18 days, the forager population turns over completely. So what that means is that foragers are replacing their entire forager population, sometimes basically twice a month, essentially, right? A little longer than that. And in all of these colonies, regardless of the number of foragers, whether it was 1,000 or 30,000, about 6.5% of foragers within a given forager population died per day. So this is quite a predictable death rate, right? And it's something one wouldn't necessarily expect across a wide range of forager population sizes and presumably colony sizes in a mosaic of different types of territories. What we can learn from this is that colonies will turn over completely about 20 times a year. These ants are active all year round. They forage the entire calendar year, and so they turn over their entire population that many times. We break this out to look at individual colonies a little bit more. Don't be scared. We can see that the number of foragers in a colony throughout the annual cycle differs markedly between different colonies. So each one of these boxes shows the number of foragers on a specific date in a specific colony. So here I have 8 colonies, and on the x-axis we have day of the year. On the y-axis we have the forager population size as estimated by mark recapture, and with 95% confidence intervals represented in the blue surrounding. What we see is that these colonies are actually all neighbors in one little neighborhood, and that for every forager that died in these colonies, colonies did really different things to replace those foragers. So although they were experiencing a pretty conserved death rate of about 6.4% of their forager population per day, on average, some colonies could replace every dead worker with 42 new workers, while other colonies could only replace each dead worker with 2 workers at their peak. Okay. Colonies with more foragers actually reproduced during the calendar year of this study. So it suggested that foraging and reproduction were tightly linked, and that the number of foragers actually affects the ability of a colony to collect food and invest in reproduction. What we calculated was that the most productive Veromssor pergandei colony in the study would produce approximately 652 successful eggs per day at a rate of 27 eggs per hour if production were equal across all hours and days of the annual cycle. Okay, but the interesting thing I think here is that there's not just that there's these extremes where some colonies are extremely productive, but the fact that these sort of low productivity colonies exist sort of in this matrix of nest territories altogether with the other ones. So we can ask, how do some colonies effectively replace so many workers while other colonies do such a poor job of it when they're both experiencing the same sort of extrinsic risks or intrinsic risks of mortality, right? And so to look at this, I sort of mapped the foraging ranges and foraging columns of all of the colonies in each of these neighborhoods across the annual cycle and looked at their total foraging range. What you see when we compare 2 colonies of vastly different productivity is that colonies like VP30 occupy a very small territory within their neighborhood. They border VP29, which is the largest colony in our study and the most productive, you know. And so on an annual scale, a small colony like VP30 would produce just 31,000 ants and 40 grams of dry ant biomass during the entire annual cycle. In contrast, its neighbor VP29 will produce about 237,000 ants and 470 grams of dry ant biomass in the same annual cycle. So why is this? Well, we can get some clues from the way that these colonies vary their investment in body size across the annual cycle. So in the 1970s, Steve Rissing observed that Veromssor pergandei colonies experience what he called a triple crunch. That is, they have a season of low seed availability, the necessity to produce workers, and concurrently to produce sexuals, and that occurs in winter. Okay, so because of this triple crunch, he observed that in some ant colonies and some Veromssor colonies, worker size was reduced following sexual production. Okay, and so when I looked at a number of colonies across the annual cycle at my field site, I found something similar. So let me orient you to what you're looking at here. This is a sort of graph showing the body size distributions, or rather the head width distributions of polymorphic workers within 4 different ant colonies across different months of the year. So this is one annual cycle, and we're just looking at 4 Veromssor pergandei neighbors from the same neighborhood. Okay, so if you just focus on the pink colony, our friend colony 29, the really big productive colony, and you follow it from March to May to July to September, you see a strong shift in the distribution of worker sizes that these colonies are producing, right? They're investing a lot more in what looks like smaller workers in May and June, and they have a sort of more normal distribution of worker sizes earlier and later in the year. Okay, but of course, all these colonies seem to be doing really different things. And so when we consider, how colonies are able to invest in maintaining worker number to maintain colony size, we can consider that they might be investing differently in worker size to do this, right? So I told you that over the annual cycle, some colonies produced a lot of workers and some colonies produced a few workers. Let's look at a colony that produced just a few workers. On the top graph there, we see the relative change in forager number on the bottom with black circles. So this is just the ratio of foragers on one date to the foragers on the previous date. So this is like a factor change in the number of foragers the colony is putting out. You can see there's very little change in forager population size in this colony. Now, we can also look at worker head width across the same cycle and at the same time points. And we see that there's not an enormous change in investment in workers of different sizes. There's a little dip there in July and August, but essentially worker size doesn't vary too much. Okay, if you want to look at that another way, you can look at this sort of joy plot at the bottom. Okay, so now let's compare to our friend VP29, the big productive colony. What we can see here is that the change in forager number is quite dramatic in the summer, right? Foragers, the forager population size increases like 11-fold between March and May. So these colonies are producing an enormous number of workers. Interestingly, in order to produce this enormous number of workers, they are reducing forager size. Okay, so if we look at the open circles, which are forager head width, you see that the peak in worker production is met by a dip in worker head width. Okay, so a really different allocation strategy. So it seems that in order to produce more workers and replace more workers, colonies produce smaller workers. When we look at this in terms of territory size, we see that colonies that produce more workers, even though they're smaller, actually monopolize much larger foraging ranges and defended territories in the neighborhoods where they live. So I'll draw your attention again to this puny VP30, which had sort of moderately sized workers all year round, had very low worker replacement, versus VP29, which produced lots and lots of workers that were very small bodied and could presumably take individual foraging trips and defend more of their territory, right? So by investing in small workers, these colonies are able to expand their territory size, sometimes even oozing into the territories of their neighbors. Now, just how many workers do colonies produce per hectare? I had to know. And so I took the dry weight of all of the annual production of workers marked and recaptured and released in the field from subsamples, and found that for a density of about 14 colonies per hectare, these ants were returning about 3.5 kilograms of dry ant biomass to the Sonoran Desert. To put that in perspective, that is equivalent to 1 wet weight coati, 7 roadrunners or 2 kangaroo rats, and even a small human infant, right? So these ants are producing a lot of biomass, and they're doing it by converting seeds into ants, okay? Now, another interesting wrinkle to this story is the fact that, there's more than one way to increase worker production in a colony, right? You could add more queens to increase sort of the egg laying rate of a colony, or you could manipulate sort of the number of queens, a number of males that a queen mates with to increase genetic diversity in a colony. And folks like Blaine Cole and Diane Warnes have shown that in genetically diverse colonies where Pogonomyrmex queens mate with many males and are super polyandrous, the foraging period of the colony is extended. So individual workers have sort of these circadian preferences for foraging, and that is dependent on patrilineal identity, right? So I was curious if the same thing might be happening here, and if perhaps the most productive colonies actually had multiple mothers or multiple fathers. Well, in fact, all the colonies in my study had a single mother, but they had about 3.8 on average male matings per queen. So these are polyandrous queens, as Sarah Helms-Cahan had already discovered. But when we map things like productivity and production of workers and plot that against the minimum observed patriline number, we can actually see an inverse relationship that's somewhat unexpected. So for colonies with fewer patrilines, we actually see a higher maximum forager population size during the year. You can also look at the total number of workers produced in the year, and this will show you the same pattern. So essentially, the more fathers a colony has, the less productive they are, and the less variation in worker size there is. Of course, this led me to ask whether worker body size could be linked to any specific patriline, and it couldn't. So this is truly something that's controlled by worker investment in feeding different larvae with different nutrition, right? So these worker body sizes are not the product of a heritable genetic difference, but they are the product of differences in nutrition and differences in feeding in the larval stage. So what could be the explanation for this inverse relationship between genetic diversity and allocation to foraging? I thought about it long and hard, and this is work I actually did with Jürgen Gadow and Bert Holdobler, and we came to the conclusion that, there may be some interesting story here where queens must mate with multiple males to find some that are genetically compatible, and if they mate with just a few males, they may strike out. If queens mate with just a few males and they strike in and they get lucky, they can be quite productive. In contrast, some queens which mate with less compatible males may be less productive, but still be able to survive and reproduce. What we see is that there's unequal sperm use by these queens in these colonies, such that usually about 44% of the workers are sired by a single male, while the other patrilines are represented in various minorities. This also led me to think that perhaps the large productive colonies, or the ones with the most foragers, are actually just older colonies. And because we see this disproportionate use of sperm by queens, that perhaps some patriline sperm donations are being depleted over the lifetime of the colony. Now, sperm depletion does happen in things like fire ants, where they actually run out of sperm after about 6 years. And this can explain sort of the death of the colony, right? For queens that mate multiply, storing a lot of sperm and maintaining a high production rate may allow the colonies to persist longer. But the fact that males may contribute unequal amounts of sperm may suggest here that the oldest colonies actually contain queens that have depleted most of their minor sperm lineages that are stored in their spermatheca. So the mystery continues, and I'll let you know if there's a resolution soon. Okay, if we think about sperm just a little more, we can see that we know that across ants that females waste about 3 sperm per fertilization event. Okay, so this is gleaned from Walter Tschinkel's work on fire ants and fertilization efficiency. If we take the same number of sperm usage and we apply it to our friend Veromssor pergandei, it suggests that an average queen would run out of sperm in about 6.5 years, based on a minimum patrilineal number of like 3.8. Okay, so that is if she maintained the same rate and level of production that she had during the course of my study. Okay, and this would be from day one after colony founding. We know, in fact, that colonies actually have an ontogeny and it takes them several years to reach reproductive size and age, and that these strategies may vary across years, right? So colonies need not invest in small workers every year. They may take sort of a gap year in terms of reproduction or in terms of territory dominance. If the lowest producing queen in our study was actually to maintain her production rate, it would take her about 34.7 years to run out of sperm with the rate of workers she's producing per day. Now, Bob Johnson estimates that these colonies live between 12 and 18 years. So these estimates aren't super far off, given that colonies have sort of that early ontogeny and potential to vary worker production on an annual scale. So just something to think about in the whole scheme of the factory, right? But what you might be wondering by now is, because body size changes so enormously across the annual cycle, how does it affect other things that go on in the ant colony, right? Selection is acting on all of these moving parts of the society and emphasizing some and not others for the benefit of reproduction, right? So if body size is important to some function in the colony, might it not matter if body size changes so radically across seasons and years? We know from Walter Tschinkel's work that ant nest architecture is extremely complex and species specific, and that ant nests sort of vary around a set of species typical traits. So here pictured are a number of Solenopsis invicta fire ant nests. These are made by different colonies. And while colony size can definitely impact the shape of a nest, there are other idiosyncrasies that are not related to the number of workers that produce the nest. So it's the interaction of different worker types in terms of age and size that can actually affect these structures. The influence of worker size has not previously been studied on natural nest architecture. So that's something I was definitely curious about when I noticed that the ratios and distribution of worker sizes across the annual cycle were so variable. Right? So nests are really important to the functioning of the colony. They are the fortress that contains the factory, right? They serve physiological functions. They can also organize labor and space. And they're the places where specifically Veromssor colonies store their enormous caches of seeds. They represent a response of the individuals in the colony to the environment, but they can be the product of intrinsic differences as well. So let's travel to the Mojave Desert, where I worked a little bit on the nest architecture of Veromssor pergandei. This is a cast of a Veromssor pergandei nest made by Walter Tschinkel, again, my PhD advisor. And it's sort of a sloppy nest, isn't it? It's got all these weird projections and discs, and is a little different than the tidy vertical nests we're most used to from work done in Florida, right? So this is a messy nest overcoming messy subterranean conditions where there's lots of stones, a dry caliche layer, and essentially no moisture. The ants are scraping away at this hard pan in order to excavate here. Nonetheless, the surface is quite beautiful, and it is a fabulous place to work. So I wanted to know what the influence of polymorphism in these Veromssor pergandei societies is on nest architecture and how it affects the structures that they build. Okay, these colonies move 1 to 4 times a year. They're actually quite diggy. They like to move often, and sort of every time it rains, they will relocate, sometimes just a little distance away, not very far. I conducted this study while working at home in Phoenix. It wasn't during a pandemic, but it was the easiest way to do it. And what I did was I asked how the interaction of different worker distributions would affect the architecture of the nests produced by those workers. And to simplify this, I created just sort of 3 very, basic treatments. I divided the distribution of workers from all the colonies I had observed into thirds, and took workers from the bottom third and the top third from numerous nests and put them into these three treatments. I took either 60 large workers, 60 small workers, or a mix of 30 large and 30 small, and allowed them to dig nests in buckets for just 48 hours. So here are sweet Veromssor digging their nest, and here I am extracting a wax cast of that nest made after 48 hours. Here you see three different treatments coming out at the same time. But in that first video, you were seeing a mix of large and small workers working together to create a nest. And so my question was, if we just simplify the worker morphology ratios, can we observe any differences in the nests that a colony builds? Okay, so the nests built by groups of small workers were unsurprisingly quite small after 48 hours, not much to see there. The disc you see at the top is actually what we call the cookie, and that's a little divot where we pour the paraffin wax to cast the nest, right? Nests made by 60 large workers are a little bit larger, but not significantly. And so one would expect that the nests made by a mixture of 30 large and 30 small would represent half of the nests made by just small workers plus half of the nests made by just large workers, right? So these are 60 workers making a nest together, and they come from two size classes. So this is sort of our expectation. When you actually put 30 small and 30 large workers together, you get something else. You get a much longer nest, okay? Something unexpected, something non-additive, which means that the interactions of these different worker types yielded a colony level trait, this extended phenotype, that was not predicted from just the sum of its parts or the sum of any one worker caste in isolation. When we look at this more closely, we can see that mixed groups of workers that included both large and small ants actually produced longer nests and excavated like a greater volume of sand than did any one size class in isolation. So these nests were much longer than expected and actually much wider than expected too. So here we can see some of these nests represented here, sort of the interesting ones. And each colony is represented by a CG number. So in the rows, we have colonies, which were divided into these three treatments: small workers only, large workers only, or a mix of large and small. Now, when I wanted to look at something else about the nest. I wanted to not just look at the size of the nest that a mixture of worker sizes created, but I wanted to look at sort of the complexity of the nest. So to do this, I represented these nests as sort of networks with nodes and branches, right? And could describe sort of the distribution of tunnels and chambers, and actually make a calculation of what we would call complexity, okay? And so what we found was that when a mix of large and small workers are placed together, they actually produced much more complex nests. You can see one of those nests at the bottom is represented by a network compared to nests made by small workers alone or large workers alone above it. Now, the large and small workers did not create any different nest features. It wasn't as though one group of workers was responsible for digging chambers, while the other was responsible for digging a turret or a tunnel. In fact, both size classes participated in the construction of all parts of the nest. However, when the ants were placed in a mixed group, they actually excavated tunnels that represented the full range of body sizes within the nest. And this is likely because in isolation, they were digging tunnels that were size specific or could accommodate their own body size. This actually suggested that large workers were working and digging at digging faces and sort of shuttling small workers off to the side, who would dig sort of these small tunnels, which would later be expanded and elaborated on by the large workers. So this is sort of an ongoing thing we're observing with video and everything to demonstrate. But there's sort of this division of labor that happens when these ants work together. And in fact, the largest workers, of course, can carry a lot more sand per trip out of the nest. They use a basket of hairs under their chin called a semaphore to carry boluses of sand. And the larger workers are sort of like really nice bulldozers and can carry a lot more sand grains per trip than the smaller ones. And in effect, when large workers are working beside small ones, we find that small workers actually don't bring sand out very much at all. They specialize exclusively on digging their small tunnels. So there's a sort of an interesting division of labor that arises. And we did this work in sand of a single particle size. When we add in sort of more complicated particle sizes like gravel and silt, we see a further division of labor between particle sizes, right? Where large workers are specializing on very large objects that small workers cannot move. So it's an interesting demonstration of the effect that seasonal distributions in body size can actually have on the nests that these ants build. And since they move frequently, we can also consider what that means for incipient colonies. Now, unlike a lot of other ants, Veromssor produce a full range of body sizes sort of after the first year of life. So they stop investing in just tiny minims, and they very quickly produce a variety of worker sizes early in the life of the colony. This could actually aid colonies in getting deeper and producing nests more quickly to get out of the sun and off that dry desert surface, which is one suggestion for why this polymorphism might develop at the beginning of the life of a colony. Okay, so we can see that body size influences territory, total annual production and colony size. It influences nest architecture, but how does it influence defense? So Veromssor pergandei is plagued by a number of very pesky theroidid spiders. Pictured here are some Euryopus, beautifully illustrated by Amanda Hale. But I want to focus today on Steatoda and Asagina, this other type of what we call false widow spider that actually seem to specialize on ants in the Sonoran Desert. So this group of spiders will actually camp outside of Veromssor pergandei foraging columns in the morning and set up a number, sometimes up to 7 web traps along the foraging routes of these ants, sometimes going directly across the large foraging column of ants. Now, while I was conducting my mark recapture studies, I noticed that there was a lot of drama going on between the ants and the spiders. And it wasn't just sort of a typical interaction where the spiders were chasing the ants or the ants were chasing the spiders. In fact, these spiders go sort of on a circuit and check the little trap webs they've set up, these sort of disposable, cheaply made webs, which they only use for the few hours that a colony forages each day. And when they're gone, ants of a large body size come over and remove those spider webs. So here's a video I took in the lab. It's probably one of the clearer ones that I have of workers participating in spider web removal. So when I looked at the size of workers that were participating in spider web removal, I think it was over 85% of them, maybe even higher, were in the largest size class of workers produced by Veromssor colonies. You can see sort of a medium-sized worker participating here in removing the spider web, but really it is the large ones that are exclusively doing this job. And when we look at why these ants are attracted to the webs that contain their sisters, we find that these sisters are actually releasing an alarm pheromone from the mandibular gland, which attracts only the large workers to them. Now, interestingly, during rescue attempts, about 6.3% of large workers get tangled and die themselves, or are eaten by a spider that returns to its web during the rescue attempt. Okay, the presence of a spider in a web did not in itself attract workers to the web and did not inspire web removal by large workers. When workers got their seeds tangled in webs, they did not remove the webs. It was only when a nestmate was captured and presumably releasing this mandibular gland pheromone, which we were able to mark on dummies and inanimate objects, that large workers would come and rescue their sisters. So they actually removed the spider web and dragged the sister back to the nest, living or dead, and removed the full complement of silk that she's been bound in by these predatory spiders. Now, why would a forager invest in saving the life of another forager? As we know, they only live 18 days. They're cheap and disposable, right? The colony turns over its foragers rapidly. Why invest in saving one, right? Well, as it turns out, if a colony were to lose just 5 foragers a day, 5 new foragers a day, to spiders, that would cost the colony about 65,000 seeds a year, assuming that the ants have 2 to 3 successful foraging trips per day. So over the 18-day tenure of a single ant and over the accumulated tenures of 5 ants lost per day over the year, there's actually a significant cost to losing ants to spiders. And this suggests one reason why workers might invest in spiderweb removal. And the fact that these large workers are actually able to manipulate and deal with these spider webs is one reason we think that perhaps small workers don't respond and invest in this activity. Now, large workers also remove seeds directly from plants. So this sort of pulling and plucking behavior is something common to this larger size class of workers within the colony anyway. And it's something that the small workers don't perform. So once again, we see another sort of difference in how body size may influence another colony level trait, for instance, defense in this species. So we can, as we discussed, workers' body size influences colony size and production, territory size. And in those cases, increasing the number of small workers you produce seems to be an adaptive strategy, and that it allows your colony to survive and maintain its size for another year. Worker size also influences nest architecture, and the interaction of different worker types seems to create deeper and more complex nests, which may be beneficial in the desert when these colonies are trying to get under the ground rapidly. And of course, worker size influences colony defense, where large workers are useful in removing spider webs and in other foraging tasks. So I think it's very interesting to consider these different aspects of the contributions to this sort of factory and production metaphor, because there seems to be a tug of war on sort of a simple single worker trait, body size, which affects these colony level outcomes. But how else can colonies optimize production? To answer this question, I want to travel to the Apalachicola National Forest in the northwestern panhandle of Florida. This is just south of Tallahassee, and talk to you about another seed harvesting ant, Pogonomyrmex badius. Okay, this is a seed chamber showing some of the collected seeds of this species. So these colonies are found mostly in sandhill habitats, but also sometimes in the flatwoods, and they produce the famous and enormous nests shown here in a cast by Walter Chinkle, right? These are some very interesting colonies because they have a single sort of reproductive event, which is coordinated with the summer solstice around June 21st each year. During the beginning of the annual cycle, which begins in March, colonies produce a mixed brood of workers and sexuals, and then they produce subsequent worker cohorts throughout the year to maintain their size. I wanted to know just how many individuals in a colony were allocated to the task of foraging and how labor was divided in these colonies across the annual cycle to determine how it would influence the production of sexuals. Okay, and so to do so, I had to again estimate the number of foragers in a colony by mark recapture. I did so with fluorescent ink, our old friend, and then also excavate the colony and completely census it in order to determine the proportion of the total colony size represented by foragers, right? So number of foragers divided by total adults equals the proportion foraging, right? Here's the census. This, of course, required a lot of digging. I did this for 55 colonies over 4 years. These nests can exceed 3.5 meters in depth, but luckily they're in very soft, buttery sand. So if you are 5'1" like me and have a short shovel, you can do this all on your own. Sometimes it necessitates the use of a ladder, which can be installed in your hole. But the results are very satisfying. What we see is that across 4 annual cycles, there is a sort of conserved pattern of allocation to foraging. So foraging begins in sort of March and April, and then climbs to a peak of about 35% in midsummer. Okay, so this peak in foraging and the proportion foraging reaches its maximum in the same way in multiple years, right? So this pattern repeats itself. This is the annual cycle of foraging. When we look at how colony size interacts with allocation to foraging, we can see that colonies of all different sizes follow the same rough pattern of allocation across the annual cycle. So here the polka dots are scaled to the size of the colonies. And if you remember back to the beginning of the talk, I told you that colonies of about 700 to 800 individuals are sexually mature. And numbers smaller than that, they're not producing sexuals. So you'll notice that some of the highest allocation of foraging takes place in very small colonies, which are sexually immature. So how does this pattern arise? It can actually arise in two separate ways: through a change in the number of foragers being allocated to the task of foraging, or it can be attributed to a change in the total number of workers in the colony as a whole. So is it a numerator or denominator problem that causes this allocation pattern? And what we see when we look at the number of foragers that individual colonies allocate across the annual cycle is that sexually mature colonies and immature colonies, of course, are doing two different things. So mature colonies, represented in black here, which invest in the production of males and queens, which will disperse to start new colonies, actually show a reduction in forager population size in the summer. At the same time, colonies that did not invest in sexual reproduction invested more in worker production and show a peak in the numerical allocation to foraging sort of in summer. Okay, so this is sort of just a difference between immature and mature colonies, which is a function of number of foragers. So the numerator in that equation when we think of proportion foraging. And this, of course, is a consequence of differential investment in worker versus sexual brood during the beginning of the annual cycle. Now, regardless of whether you're looking at a mature or immature colony, we can see there's a very conserved and strong relationship between foragers in the colony and the number of larvae that colony produce. In fact, colonies contain approximately 1.6 foragers per larva across the annual cycle, regardless of forager number or forager population size or colony size. So this is a very strict relationship between the number of ants that go out to collect food and the number of larvae that they can produce, that the colony can produce based on the income, the seeds and insect prey that these colonies collect. Okay, so how is this relationship controlled? Is this a flexible relationship where allocation may differ based on demand? So does the number of larvae in a colony actually drive allocation to foraging? Or perhaps we can think of the fact that mortality rate and the loss of foragers might drive allocation to foraging. What is driving this proportion that is so nicely conserved across colonies in this population? Well, the first thing one can do is actually remove half of the forager population from a number of colonies, right? So it's easy to estimate how many foragers are there. We get, in this case, more than 96% of them back on day 2. And so on day 2 of the mark recapture event, you can simply keep 50% of the estimated total forager population. If you return a week later to check on whether foragers have been reallocated to this task to replace those that you took away, you find that the forager population is still missing half of itself. So they did not replace the foragers that are taken after 7 days. They do not reallocate, right? So it's pretty inflexible. And at the same time, the expected ratio between larvae and foragers in a nest is conserved. So when we dig up these nests, we find that there aren't sort of double the larvae. There's just the right amount of larvae for the number of foragers that still remain, suggesting that the workers either cull and feed out the larvae or the larvae die of starvation. Presumably they feed them to other larvae. Okay, well, let's do the inverse of this. What if we increase demand for foragers by increasing the number of larvae in a colony? What would that do to allocation to foraging? Can we tinker with it? Can we get it to change from this strict seasonal cycle? When you add or double the larval population in the colony by giving a colony conspecific larvae, which they readily accept, the colony does not increase allocation to foraging over a week's time, and actually over 2 weeks' time either. And when you dig those colonies up, you find that there's sort of a equal proportion of the donated larvae from conspecifics and the host colony's larvae that were originally there, suggesting that the excess larvae were indiscriminately culled or fed out to other individuals in the larval population. So these colonies do not reallocate and increase their forager allocation even when they have more demand for foraging. And this is in the field, mind you. So these colonies are taking these larvae in, which we can see from the marks, and putting them in their brood piles, caring for them presumably, and simply not reallocating foragers to bring in more food. Okay, well, can we change anything about the foragers themselves? Can we get them to revert to earlier labor roles? So if you're an ant biologist like me, you know that there's a strong relationship between worker age and body fat content within ants. So as ants age, they lose body fat. Okay, so foragers measured in this species on average, I saw had about 3.5% body fat only. So they were extremely depleted in lipids, and which suggests that they were sort of towards the end of their life and functionality. Well, if you actually pin colonies and prevent foragers from going out to forage and then feed those colonies, you can in fact increase the proportion fat that you find within the foragers. So you can reverse their body condition to that of a middle-aged worker by simply by feeding them mealyworms and sugar water, okay? And so I was able to get foragers to return to an earlier body condition and then remove the pin and see who was recruitable to foraging and who would participate in foraging after a 20-day period. What I found was that when foragers were prevented from foraging and either fed or starved, they gained weight and still maintained their role as a forager. We could not get them to revert to different labor roles or return to work in the nest. They were still completely recruitable to the job of foraging even after 20 days. Okay. Now let's do something really crazy. What if we were to increase the forager population size? Would the forager population size be at all flexible? Would some of the workers that were already there or those that we added actually return to interior labor roles and leave the forager population because there were simply too many foragers? So what I did here was take those colonies like before and remove 50% of the forager population. After 20 days, for those ants that were still surviving, about 40%, I was able to return that to the host colony. They readily accepted back their own foragers, right? Over this time period, the colonies over 30 days actually had recovered their missing forager population. You can see this very slow recovery and the fact that the colonies will return to the population size of foragers they had before the removal over a longer period of time. Certainly not within a day or within a week, about 30 days. If you return the lost foragers then at that time, in fact, they retain those individuals as foragers, and those individuals will keep working as foragers until they die. So they do not revert to earlier labor roles, nor do they cause the reversion of the current forager population to interior tasks. Okay, so this is starting to suggest that there might be something about colony demography that's driving allocation to labor, right? And so to study this, I took 11 colonies and actually divided individuals by their cuticle color, which often corresponds to age, such that very young workers are light colored and old workers are dark colored. I marked them with these wire belts and also with fluorescent printer's ink, and then returned them to the field by making facsimile colonies out of ice. So you can actually prevent the ants from having to do any work of digging a new nest, and you can recreate their own nest by freezing ice cubes in the shape of ant chambers, and then burying those chambers below the surface, allowing them to melt, and then putting the ants back into the colony that you created for them, the nest that you created for them. And I did this so that I could mark all individuals in the nest and not have them lose body fat or experience the costs of re-excavating a nest. And to just to show you how well this works, in the middle here, you see a photograph of a real nest for a small Pogonomyrmex colony. And on the left, you can see one of my ice nests that I made using ice cube trays made out of copper strung together by Tygon tubing. And so this ice nest, when it melts, creates spaces under the ground, which you can either cast or put a new ant colony into, or even the one that you just marked, okay? So when I've put fully wire marked Pogonomyrmex colonies back into their own territory in an ice nest that precisely resembled the nest they came from, here are these lovely ladies with their belts, I found that they then became foragers at sort of different rates depending on the season when they were marked or the color of the cuticle they had at the time of the mark. And so you can actually monitor the forager population on the surface by catching workers actually foraging for seeds, and look at the color of the belt they're wearing, which indicates the sort of age or color of cuticle they had at the time of marking. And so when I did this, I found that individuals that were closing from their pupae in June and July, the first cohort of the year, actually developed rapidly alongside males and female alates to become foragers at just 43 days of age. So they became foragers very, very quickly. In contrast, those workers that appeared in August and October or closed from their pupae at that time took sometimes up to 280 days to enter the forager population. So there are two extraordinarily different rates of aging going on here in the colony, which affects when individuals enter the role of forager. Okay, now to understand this a little better, we need to think about the diet of these ants and what they eat on an annual cycle. So this is work I did again with Walter Chinkel for my dissertation. And what we found was that, these colonies are storing many, many kilograms of seeds, but most of the seeds in the colony are actually quite large and too large for the ants to mechanically process and open. So if you look at this graph here on the far right-hand side, you can see a couple interesting seeds, Ceanopuntia seed, some euphorbs, the spurge nettle, and then also smilax, everybody's least favorite vine in Florida. These seeds are collected in extreme abundance by the ants. They love them, but they cannot physically open them given the cross-sectional area of their mandibular muscles and the action of their mandibles. So what do? We actually find that these ants wait for these large seeds to germinate. And so they're at the mercy of the germination schedule of the seeds when they're trying to access the pulp inside. So in essence, Pogonomyrmex badius collects thousands and thousands of seeds, and it can very rapidly and nicely access small seeds whenever it needs and wants them. The larger seeds, of course, must germinate on their own schedule, which we simulated in the lab in different temperature regimes and in the field in divided chambers in subterranean nests. The ants prefer to eat germinating seeds. It begins the malting process, converting starch to sugars, and they seem to store these for the purpose of sort of delayed benefits of seed collection. And so when do seeds begin to pay off? When we think about the diet of ants in different seasons in this species, I did a stable isotope analysis to see if seeds were being eaten more in one season or another. What I found was that essentially workers created alongside sexuals were fed a very high protein diet. It was likely the result of mostly insect protein, and they were fed on like a high trophic level, right? So their delta N15 suggested they were close to being a full predator. They were likely also eating trophic eggs laid by their larger sisters. In autumn, when the slow developing phenotype workers were produced, the diet of those workers was more biased towards carbohydrates and specifically seeds in the environment where they were found, which suggests that after winter, either there's no seeds germinating immediately, or the small seeds are depleted, and the germination schedule that really heats up in the autumn in Tallahassee area is beneficial to these ants because they can begin to access a number of seeds concurrently to produce that large, slow developing, and actually very fat cohort of pre-winter workers that begins in August. So what does this mean for the colony as a factory? It means that workers of the same cuticle color and in the same labor role are of vastly different chronological ages at different times in the annual cycle. Old workers are passed by hundreds of sisters in that new cohort in any given focal year. So individuals born the year prior are like passed in terms of labor allocation and job by their younger sisters born alongside sexuals. Okay, but how do death rates differ? In fact, like Veromssor pergandei, there's a pretty consistent mortality rate within any given forager population, such that about 3.7% of foragers die per day in a forager population of any size, as shown here by the different sized bubbles. So we're thinking about a group of foragers from a colony and the number surviving or the percent surviving over time. No foragers are really living longer than 25 days. Okay, so that's when complete turnover occurs. The number of days an individual spends as a forager is also largely conserved across the annual cycle. It begins to increase during November when foraging is decreasing because these ants do overwinter. And the lifespan of a forager, once it enters that role, does not differ between those fast developing and slow developing phenotype workers. So why might this be? Well, what we see is that there is a seasonal decline in forager replacement, even though dead foragers are dying across the annual cycle. So we look at the black bubbles here, we see the number of foragers dead in any given colony forager population at these different time points, and then the number of foragers replaced. And we see that replacement just drops dramatically in September and October. So although the colony is full of these young light-colored workers, they are the slow developing phenotype. And although they are there and recruitable to foraging, they are not becoming foragers, even though the demand for foragers is still high because death rate is sustained. So this suggests that demography really is driving the allocation to labor in this species, because a reduction in foragers, either natural or simulated by experiment, does not induct new young workers into the role of forager over time. But we can interrogate this further, right? We can look at really the relationship between extrinsic mortality and forager lifespan to see if we can tinker with that longevity and ultimately the allocation of foraging by the colony. So what would happen if we got foragers to live longer than that 25 days? What would happen? What would happen if we removed extrinsic sources of mortality? Well, we can actually do this with that simple pinning experiment. We can prevent foragers from foraging. We can make them stay close to their nests so they can't leave and get eaten. And we can feed them there or actually starve them there in their nests. We can run concurrently a bunch of control colonies in pins with doors so the foragers can open to make sure there's no effect of the pin on survival of foragers. And what we find is that whether you're looking at a control or a fed or enclosed and starved colony, forager population size is really maintained across the 20 days of the study. Okay, so if you have 500 workers on day 1, you're going to have about 500 foragers on day 20, regardless of what treatment you're in. Okay, but what we find is that the way that this occurs is a little different, such that controls consist mostly of new workers, new foragers entering the forager population, with only some of the old foragers from the first day of the study remaining there. In contrast, those colonies which were protected from extrinsic sources of mortality still contain many of the old foragers that were there at the beginning of the experiment and just a few new foragers. Okay, so although both of these treatments and the control maintain the number of foragers over time, they're doing so in different ways. What does this mean? This means that the increased survival of workers in colonies that are pinned and protected from extrinsic sources of mortality, that increased survival actually suppresses the development of younger sisters and prevents them from entering the role of forager. Okay, so this is like a unidirectional control system, because as I've demonstrated, workers do not reallocate to become foragers when there's a gap or demand for foraging, but the increased survival of foragers actually suppresses the movement of new workers into that job, allowing those younger workers to stay in the colony and continue to focus on in-nest tasks like brood care. And this is at the heart of how colonies grow. We want to think about this in realistic terms. We can think about extrinsic sources of mortality in terms of conspecifics, right? So if we were to enclose all the neighbors of a colony and compare forager survival in those focal colonies to colonies with all of their neighbors exposed in foraging, we find sort of a similar pattern to when we enclose a focal colony and feed or starve it. So the presence of neighbors actually causes the death and probably truncates the lifespan of a lot of foragers. So the actual and realized lifespan of foragers are pretty significantly different. When neighbors are enclosed and sort of removed from the foraging area, the foragers in a focal colony will survive much longer than expected, much longer than 25 days, sometimes close to 80 days. So it suggests that the lifespan of a forager is not sort of paced on sort of an intrinsic rate, which would be controlled by some physiological senescence, okay? We know that neighboring foragers fight a lot. Here's one with the head of a conspecific clipped onto her waist, right? And in fact, we can see that neighbors are a significant source of mortality and probably a predictable source of mortality for these colonies as they produce new workers each year. Now, this is just sort of beginning. I haven't done too many replicates of this, but when you look at the number of new young workers, so callows, larvae, and pupae at the beginning of the season in colonies with and without neighbors, you find that the investment in new workers is much greater. So colonies actually grow when foragers survive longer. And this is what's at the heart of the factory of the Pogonomyrmex ant colony, right? This fact that forager longevity causes a suppression of worker development and is only flexible in one direction. Now, why wouldn't colonies want to replace foragers that are lost? In fact, in many Pogonomyrmex colonies, species rather, there are predators that will prey on foragers for hours a day. Horned lizards and things of that nature will completely deplete the forager population. And in my mind, it doesn't behoove a colony to keep hemorrhaging workers to replace those foragers which are being rapidly eaten by a lizard. So there may be some benefits to this restraint and this inflexibility in an environment where the forager population could potentially be depleted by a sort of random event of that nature. I think I'll skip ahead because we are very short on time. In fact, it's 9 o'clock. So I will just give you maybe a 5-minute introduction to a new project, and then I will set you all free. But I will skip ahead. These ants do some amazing things with bioturbation, right? And the movement of seeds. But one of my main interests that I haven't talked about yet is the relationship between ants and their myrmecophiles, okay? So of course, I've given you the whole sort of economic equation of these ant colonies, but there are other little factors that can affect the way that a colony produces brood or allocates labor, right? There can be costs in the nest. And of course, there are many types of myrmecophiles, or in this case, parasites, which will eat the food and brood of their hosts. So this is a new species of beetle. This is an alloculid beetle that I found living in nests of Pogonomyrmex badius. And these are really interesting beetles because the larvae actually live in these top chambers of the nest, and they make little burrows in these depots where incoming foragers drop off seeds and insect protein. Now, the insects are shuttled down the nest in a series of caching steps and then delivered to the storage chambers and brood that are waiting to eat them. The beetles take advantage of this caching behavior that the ants perform. They also move sand up in a series of steps. And they actually are able to eat, most of the insect protein that comes into a colony before ant larvae ever get a chance to get a taste of it. So I studied this by dyeing beetles with methylene blue and rhodamine B, and actually giving them to banids that were biting me dyed with these food dyes. Okay. So there's a cost to hosting myrmecophiles, and the nature of an ant colony in terms of its worker number and worker size can all affect the types of myrmecophiles that live and thrive there. And so I want to tell you about just briefly a new project. I've been working on a book with Bert Holdobler called The Guests of Ants, and it's coming out in July of 2022, so in just a few short months. And the book is about how myrmecophiles interact with their hosts. And so we explore this from a variety of different levels. We talk about internal and external parasites, but also mutualists, predators, mimics, and sort of the parasites of the social systems of ant colonies. So here you see an antinophorus mite initiating trophallaxis with its host. These Lyphra caterpillars that cruise across the Ecophila colonies and eat their brood. They've been described as a creeping soul. Of course, wonderful Batesian mimics that benefit from running with and looking like ants. And among them, these beautiful spiders, which mimic a variety of ant species. Some of my favorite guests are phoretic. They are riding on ants to disperse to new locations. including the world's smallest cockroach, Adaphylla, right? So this is a scientific examination of socially parasitic invaders from butterflies to bacteria that survive and thrive by exploiting the communication systems of ant societies. And for those of you that have a soft spot for vertebrates, I do too. And so there's a whole chapter devoted to a number of creatures that have backbones that actually make their livings off of ants, either in the colony or on the surface, including a species of frog which deposits its tadpoles in the nests of Atta. Okay, so we are very short on time, but I'll just mention that my great interest is in ant crickets, and especially the North American ant crickets, which vary enormously in body size depending on the host that they're found with. So I've been looking at a number of species, specifically Myrmecophiles manni, which is the desert ant cricket, and now Myrmecophiles pergandei, which lives here in Massachusetts, and looking at the relationship between host size, host ant body size, and cricket size. We find there's like a very strong relationship with a few deviations which relate strongly to the diet of the ants that are hosting these crickets. So in this brief video taken by my master's student, Jen Ingram, we're going to see one of the ways that crickets exploit their ant hosts. Of course, they can very nicely interrupt trophallaxis bouts or regurgitated food sharing between host ants, as you'll see this cricket do now. And this is one way that they can get enough food to molt and grow. And it may be the basis of why some crickets are larger than others, depending on the body size of their host. In species that don't trophallax, like Pogonomyrmex shown here, we see they fall far below the regression line, and crickets are much smaller than you would expect them to be based on the size of their host. So there's likely a very interesting dietary component that actually affects why these crickets match the size of their host so well. And in fact, you can see this between species, where the largest ant cricket in the world can actually be found with the largest ant in the world. I'm working with Jeffrey Sosa Calvo to sort of make sure that we're not seeing cryptic species here. And the body size is really something that's plastic. We can induce it by cross-fostering, but in the field, when we sample crickets from different field sites and different populations, we find that within the same clade, these crickets are exploiting multiple hosts. So closely related crickets have multiple ant hosts, shown here by different colors. And crickets of vastly different size are found within the same clade with those different hosts. So it does suggest that this is body size plasticity and not a case of sort of cryptic speciation. Regardless of body size, these crickets all lay the same sized eggs, which is an interesting life history mystery. So I think that I will end there by just saying again that, an ant colony is a factory within a fortress, and there are some little production costs that come in the form of mecophiles. But it is really fascinating to see all of these moving parts, from workers to labor groups to extrinsic sources of mortality, that all affect a colony's ability to survive perennially and replace the workers that it loses, right? So I hope you will continue to think about the ants below your feet. And in fact, some of my students pictured here, Roman Meneghini, I have been working on a local ant, Phydola prolifera, and the fact that it actually mills and processes seeds during the peak of winter cold. So these ants are winter seed millers. So hopefully you'll see Roman around here another time. I will end there, and thank you for your attention. I really appreciate the opportunity to speak to you today, and hope that I can see some of you around once the pandemic calms down. And I'd love to see some of you